I'm Bob Tranquillo at the University of Minnesota, and I'm here to tell you that RMM has been quite significant in getting our technology translated down to a company in Stillwater called Vasculine. And that happened in a couple of ways through 2 RMM grants. One was with the Senior Research Associate in my lab, Zishan Sidain, who helped to take our technology of making tissue tubes in the lab from skin cells to a GMP production facility on the St. Paul campus at Minnesota, which led to the licensing by Vasculine, because Vasculine could see from that RMM grant that we had developed the ability to make it under GMP conditions, not just in my lab, which of course is important for real-world commercialization.
Vasculine was started on a fundamental premise of making vascular products. And some of those products were, one of them was a bypass graft. And frankly, we were struggling. It was difficult to get something that looked appealing enough to get into that product development phase. But then we were introduced into the U of M folks. So Professor Tranquillo, Dr. Sidain, and we were able to secure a couple of their products. And when we ran those side to side against our own materials, it was a slam dunk. It was, hey, look, if we're gonna make this product, it doesn't make sense to continue down our path. It makes sense to license this technology and bring it to that level. So had they not been able to provide us some materials for that evaluation, had they not been able to spend the last several years developing their manufacturing to get them that product, this would have never happened. So I think the amount of information that had been gathered by the university from the grants previous to ours had all contributed to where we're at today.
My project is making a better model for studying human immune response. We aim to genetically engineer a humanized pig model that carries human hematopoietic cytokine. If we'll make this humanized pig model, this could be the new gold standard of immune research or immunology research.
So the education program that's currently funded by Regenerative Medicine Minnesota is a phenomenal opportunity for us to enable medical students and graduate students to take part in a course to educate them on what are regenerative medicine principles and apply it to clinical practice. We've currently been fortunate to have funding for the last 4 years, and through this grant we've educated over 200 medical students as well as graduate students, visiting research fellows, and current physicians in training on what it means to be trained in regenerative medicine and surgery. They've taken these principles as first-year medical students, gone on to train longitudinally across their 2nd and 3rd years, as well as their 4th-year clinical electives and residency electives, and really gotten to build a community of resident physicians as well as practicing medical students, graduate students, and research fellows that are interested in regenerative medicine that are our next generation workforce. The goal is to really position Minnesota as the first state in our nation that's training every single medical graduate in regenerative medicine. So regenerative medicine is the future. It is really incorporating into all aspects of medical and surgical specialties. It's now time, and we've encouraged our medical schools to incorporate these lexicon into their curriculum, and we are able to now incorporate and adopt these different principles to practice at Mayo Clinic as well as University of Minnesota. Grant funding from Regenerative Medicine Minnesota is very important for us to sustain our education program in order to build a next generation workforce that are training in regenerative sciences, as well as retain these workforce in Minnesota to help adopt and deliver future regenerative principles.
So we have a couple different educational programs that are funded by the Regenerative Medicine Minnesota grant. One of them is a series of Explore STEM day camps that we offer through the summer for 6th through 9th grade students.
The ultimate goal of these educational programs is to support underrepresented students in the sciences. And so if we can introduce STEM as a realistic career for students across the state of Minnesota, we are meeting our goal.
Grant funding for our programs is essential. Especially when we're trying to offer programs that are free to little cost, so that we can reach these underserved, underrepresented populations that have these barriers, many of which are a financial barrier. We can't run these programs without that support of the grant. And the Regenerative Medicine Minnesota grant has been, over the last 5 years, has been key and very essential to the work that we do with these Explore STEM programs and making an impact with these youth, because without that grant, we wouldn't be able to offer these programs.
Many investigators along the way have been testing the potential of cell-based therapies for diabetic kidney disease. And in our own lab, we were doing these same things in renovascular disease. Now, what we want to do, though, is bring that from the bench, where we're looking at animal models, to the bedside. The goal now was to see if we give individuals cells from themselves, will they function appropriately? And the studies to support this in diabetic kidney disease were lacking. So that's what we spent the last few years testing, the cell function and viability of adipose-derived mesenchymal stromal cells in individuals with diabetic kidney disease. Thanks to Regenerative Medicine Minnesota, we will now be conducting the very first clinical trial testing the safety and efficacy signals derived from cells coming from the patient, the individual themselves, and given back to them to treat diabetic kidney disease.
The ultimate goal of our research project is to cure diabetes. There are a lot of different parts of that, and our research focuses on one little part of that, and that's the ability to provide an unlimited supply of islets. Those are the insulin-secreting cells that can cure diabetes. The grant from Regenerative Medicine Minnesota has allowed me and my collaborators to start those first experiments, to demonstrate those first successes, which will let us apply for other grants and develop a long-term research initiative to really solve this important problem.
So for this project, we're using larval zebrafish as an animal model to study regeneration and functional recovery following spinal cord injury. There are two key advantages to using zebrafish for this type of research. First, humans and mammals have a very low capacity to regenerate nervous tissue. In contrast to that, zebrafish can regenerate many different parts of the body, including the nervous system. Ultimately, the goal of this project is to identify signaling molecules that might impact the regenerative response. And hopefully these signals can be targeted for the development of clinical treatments for patients with spinal cord injury.
So this project is to develop new off-the-shelf vessels for use in coronary artery bypass grafts. Currently, the way that that's treated is to harvest another vessel from a different site in the patient's own body and use that as a bridge, essentially, from one of the main vessels in the body to beyond the obstruction so that we can restore blood supply to the heart. Having a vessel that surgeons could just take off the shelf and put into patients would alleviate an awful lot of problems and also speed up the procedures. Using the grant from Regenerative Medicine Minnesota, we intend to continue our research into the use of animal tissues in human patients and define both the barriers to doing that and methods to overcome those barriers. Additionally, we expect to be able to define truly regenerative processes that will turn these materials into the patient's own tissue over time.
We hope to provide a pathway to have clinicians, your retina doctor, be able to find out what kind of drugs are best for you specifically. And what we're doing is using induced pluripotent stem cells that we've made into retinal pigment epithelial cells. And we take a little biopsy of the conjunctiva, which is the white part of the eye. And we grow those cells in a dish and then make those cells iPS cells, and then finally RPE. The ultimate goal of our research project is to find a cure for the dry form of AMD, and also to help us lead towards personalized medicine for treating the disease.
My research project is on the role of smoking in causing chronic obstructive pulmonary disease, or COPD, as it is also known as emphysema. So what we want to do is see if we can use novel methods to treat emphysema. This grant would allow me to actually probably bring forth a novel treatment option for COPD and emphysema.
Our research project is about dissecting the molecular mechanisms of how red blood cells are made from progenitor cells. Specifically, we focus on to look for the molecular mechanisms controlling a specific process during the red cell production. That's how red blood cells lose the nuclease during the red cell differentiation. The ultimate goal of my research project is to identify the fundamental mechanisms of how red blood cells lost the nuclease during the differentiation, and then activate these nuclear loss programs in malignant cells, such as leukemia cells and lymphoma cells, to kill this kind of cancer cells.
In Minnesota itself, 18% of the population is dying every year due to heart diseases. And our goal is to repair the heart and recover the heart function. The ultimate goal of my research project is to find a molecule, a combination of molecules that can be used to treat heart failure patients.
So this project is really designed to determine whether a series of antibodies that we've developed here at Mayo Clinic, regenerative antibodies, can be translated into spinal cord injury. So our overall hypothesis is that if we can augment these reparative antibodies to injury patients and to disease, that we could have a dramatic effect upon repair and recovery.
The main goal of my research project is to identify molecular factors that promote neuronal regeneration, so that we hope to prevent or improve cognitive function in chemotherapy-induced chemobrain. So our ultimate goal is to develop effective drug for chemobrain and improve quality of life for cancer survivors.
So regenerative medicine involves the repair or replacement of damaged or diseased tissue. So in sports medicine or orthopedic medicine, a lot of tissues that are damaged are things like tendons or joints, so that can be arthritis or tendinitis. The purpose of our project is to take platelet-rich plasma and try to purify it by removing some of these things that can either cause inflammation or can cause catabolism, where it breaks down those tissues. And therefore, you'd have a better product to try to treat orthopedic and sports medicine conditions more effectively. The grant from RMM allowed me to pursue my dream of identifying a new regenerative medicine product that can be used to treat people who otherwise have failed other non-operative measures.
Thank you, Regenerative Medicine Minnesota.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you, Regenerative Medicine Minnesota and State of Minnesota, for funding these awesome programs.
Thank you.
Thank you.
Thank you.
Thank you to State of Minnesota.
There are many other people as well, and we can't do these projects unless a lot of people come together for a common project.
I'd like to thank Regenerative Medicine Minnesota for allowing me to pursue my dream.
Thank you to RMM for granting us this opportunity.
In addition, we have to thank our colleagues, our friends, and our families that support the sacrifices we make to bring the science forward. Thank you.